So it's my great pleasure to introduce Dr. Aaron Viny, who's giving the first of our talks in this series this year. So welcome to the new school year. So Aaron and I have known each other some while, and actually Russell, who's a PhD student of mine, is not here today because of his housing hassles, has been working quite closely with you, I think, recently. So Aaron is an assistant professor of medicine at Columbia, and he's a hematologist and medical oncologist. He's also a physician scientist, so he practices medicine and does science. He focuses on the study of clonal hematopoiesis, myelodysplastic syndrome, and acute myeloid leukemias. And he does lots of genomics, and I think he's going to tell us something about it now. So it's great to have you. There's 15 people online, I think, so we're sort of split down the middle.
Wonderful.
And here's what we're going to hear about.
Great. Well, Simon, thank you so much for inviting me to come speak. It's a huge honor and really exciting, especially on the 5th anniversary of the IICD. And I think the potential for how the mathematics, biostatistical, and computational approaches to really deconvolve how cells make volitional changes in transcriptional orchestration is ripe for new discovery, and integration with the medical center, the physicians, the scientists, and the computational biologists. Together, I think we're really going to make significant headway into a wide variety of cancers, which so desperately need our help in providing better treatment. Let's try that. Okay, so I have one relevant financial relationship to disclose, which is I'm on the scientific advisory board for ARIMA Genomics. I will be showing some high-C data during this talk, and that data was generated using ARIMA products. So I want to give a little bit of a clinical background, and I won't belabor this too much, but there's a really important feature that I want to emphasize when thinking about blood cancers, and that acute leukemia is inherently a disease of stem cells. And in thinking about the 3 diseases that Simon mentioned that we study, clonal hematopoiesis, myelodysplasia, and acute leukemia, specifically for the latter 2, these diseases differ really exclusively on the amount of disease burden observed by the pathologist when they look in the bone marrow biopsy specimen. But intrinsically, these are the same disease on the same cells. These are conditions of abnormal hematopoietic stem cells that are ineffective and inefficient in their ability to make the blood cells that we need to survive. The red blood cells that carry oxygen, the white blood cells that fight infection, the platelets that clot the blood. A striking number is that every single second, your bone marrow right now is making 2 million red blood cells. It's a phenomenal factory of cellular elements. And not only does it need to do that, it needs to do it over your entire lifetime. So that same stem cell not only needs to be able to provide a factory and a repository for these cells, but it also has to have both intact and coordinated and regulated self-renewal. When that is perturbed, that leads to these disorders. And so these both have a defective capability to generate and create these same blood cells that we need. But importantly here, if you look, and my pointer is unfortunately not able to magnify this, which is why I got this fancy pointer. If you look at this nucleus here, it just looks weird, right? And you can see that the DNA inside this cell is creating these little kind of blebs of kind of lacy patterns with kind of more coalesced DNA in there. It doesn't look like a normal stem cell. It doesn't look like a normal nucleus. At its purest form, this is something pathologists 100 years ago were able to describe, and they call it these megaloblastoid features. Well, this is abnormal chromatin. This is abnormal chromatin in a cell. We can see it with a light microscope. And now we have the capability to look at it with finer resolution using some of the new technologies and understand what is actually going on. So going back for a second, there's this disease called myelodysplastic syndrome. And when it starts to amplify, it becomes acute myeloid leukemia. Now, there are some leukemias that people don't go through that same MDS to AML phase. They just go right to leukemia. There's certain gene mutations that are necessary and sufficient to just be leukemia. This disease is still bad. I don't want to make it sound like leukemia when it just arises de novo is easy to treat, but it is slightly better with a 5-year overall survival of about 45 to 55%. That's here in the blue right there. Now, of course, there's patients who have other types of cancer, breast cancer, colon cancer, who get just incredible amounts of poisons and chemotherapies to kill the rapidly dividing cancer cells. And they do damage the healthy cells too, but they damage them at a slower and lesser amount than the actual cancer, which is why the patients live longer, which is great. But if they live long enough, some of these patients will ultimately get leukemia from the DNA damage that's here in green. And those patients do rather worse than the people who just get a leukemia. But the people who do the worst, the people who have myelodysplasia or myelodysplastic syndrome before they get leukemia, that's here in this, it's supposed to be yellow, but it's not translating all that well. And these patients have a median overall survival of 7 months. That hasn't changed in the last decades. And the reality is that there is a difference whether you've had a slow acquisition of certain mutations, or if you have the one gatekeeper mutation right out of the gate, and that changes based on the genomic landscape. So I'm not telling you that these are exactly apples to apples comparisons, but typically the last mutation is roughly the same. FLT3 or NRAS or NPM1, those typically are these AML defining mutations, but there's these other mutations that live and predominate in this MDS phase. And these are dynamically, these are specifically mutations that affect chromatin. And so in that, learning about hematopoiesis as a hematologist, giants in the field, Dan Tenon and Ken Zaret, have described hematopoietic specific transcription factors, and they turn on the right genes, right? They turn on the genes you need to be a red blood cell. If you're going to be a red blood cell, you better express hemoglobin. Otherwise, you just aren't a red blood cell. If you're going to be a myeloid cell, you better express myeloperoxidase. Otherwise, you're just not going to be a myeloid cell. And so these transcription factors help to define that. And so thinking about how you may have approached your transcriptional RNA-seq data, you may have tried to bin the genes that are upregulated or downregulated to targets of some of these transcription factors. And thinking the transcription factors alone can independently produce one of the 32 different kinds of blood cells that you have is a little perplexing, because the same transcription factor, PU.1, is important for myelopoiesis as it is also for lymphopoiesis. So how can the same transcription factor turn on distinct genes in one gene population and not in another gene population? And we think that's for chromatin architecture. That at its simplest form, one of the epigenetic principles that is underlying a cell is what areas of the genome are open and accessible, and what areas of the genome are heterochromatinized, tightly wound around histones, and repressed in a manner that that transcription factor simply can't access it. And so in a schematic that I'll kind of go through over the course of the talk, allowing the transcription factor to bind requires open chromatin. It requires the promoter to engage its transcription factor and thus turn on a specific program that transcription factor is intended to turn on. And so how this three-dimensional chromatin is arranged is comprised largely of an insulator protein called CTCF that binds to one of 300,000+ motifs across the genome and produces insulation when bound to a loop complex called cohesin to promote insulation and segregation of genomic neighborhoods. Here you can see at the highest level chromosomal territories. Chromosomal territories where they segregate these chromosomes. And interestingly, the acrocentric chromosomes, which have large amounts of ribosomal DNA, all have territories that overlap into a subnuclear structure, which we all learned in about 3rd grade, called the nucleolus. So that structure is intensely regulated. It's not just a random ball of 6 feet of DNA yarn. And how each chromosome is then subsequently organized are into these topologically associated domains, called such because in IC data, which I'll show, there are regions of the chromosome which form what look like triangles at diagonals, or the other kind of term for what a TAD is. And then within those TADs, are many promoters, many enhancers, which are all kind of co-regulated. And so think about your favorite gene cluster. Think about the globin locus. Think about the MS4 locus or the Hox locus. You have multiple different isoforms, which all need to be regulated in a concert with one another, where certain Hox genes get upregulated, certain Hox genes get downregulated, and the timing of that needs to be precise. Well, it better all be in the same tag. And then at the loop level, you have CTCF and cohesin, which it has to be CTCF in the right orientation of N to C to N, because cohesin will only tether and bind to the N terminus of CTCF to provide a stable loop, which then facilitates that promoter enhancer interaction. So I told you that the epigenetic chromatin regulating genes are frequently mutated in MDS and AML that arose from an MDS, and here's the data to prove that. And when we think about the worst leukemias that we've got, the ones that we really need better treatments for, this up here in this horizontal histogram is the supplementary data from the genomic classification of AML from Ellie Papamanuel, and showing that in yellow and in orange are the long-term survivors. One of the worst leukemias we have is MLL rearranged leukemia or complex cytogenetics P53, which I don't have on here. But we're talking, these are patients that have very, very few single-digit percentages of long-term survivors. And the chromatin modified group, which cohesin mutations are an obligate member of, do just as poorly. And so these regulators are influencing not only the genes that can be turned on, but that intrinsic cell identity of not only what genes are on, but what genes potentially could be turned on from that cell. And so this is going to the from the hopefully provocative title of a operator switchboard of how does a cell make the decision of which of the 22,000 genes in the genome A are on, and B, which ones have the potential to be on to prevent ectopic expression of certain genes that really have no business being turned on. So to answer this question, we took this experimentally to say what would happen both in patients and in mouse models if we delete the gene STAG2. And so we took a conditional knockout allele. So these mice live into adulthood, and then conditionally, like in patients, they have a somatic mutation of STAG2, thus losing cohesin and thus losing some of the key chromatin architectural regulators. It was hypothesized that cohesin's main role in cancer is to align and stabilize sister chromatids during metaphase, and that by losing cohesin, chromosomes just fly all over the place and you get aneuploidy. Well, that's not what happened. One is that's not what happened in mice, and two is that's not what happened in patients. There were no abnormalities of chromosomal copy number or cytogenetic abnormalities in cohesin mutant patients compared to the field. And in doing so, really highlighted that this is purely a transcriptional effect and a transcriptional dynamic effect during differentiation. So in the mouse, here in blue as compared to red is the STAG2 knockout mouse compared to a wild type control. In purple and in green is the same, but for STAG1, there's a second type of cohesin. So cohesin is the 3-protein ring structure, but then it's bound to either STAG1 or STAG2. This changes several molecular features of cohesin. One is that STAG1 has higher residence time and is more resistant to the loop being opened by a complex called WAPL. Second is that the STAG1 complex moves slower and is much more stable. And so it enriches at those boundaries of the genomic neighborhoods at the tags. But these short-range loops use a specific and different cohesin STAG2. And it binds and it unbinds and it binds and it unbinds in order to create this dynamism. And so what did we see? We saw when you knock out STAG2, an enhancement of your myeloid cells in the blood, an enhancement of your myeloid cells in the blood, and almost complete loss of your B cells, which is quite striking. By contrast, nothing changed when we knocked out STAG2, because everything that STAG1 can do, STAG2 can also do, but everything that STAG2 could do, STAG1 can only do some of it. So looking transcriptionally here, we first started by taking the stem progenitors together and doing bulk RNA-seq, and that was helpful to an effect. It told us that all of these different lineage prime cells have a commonality of a decreased functional output of their lineage commitments and cell fate specification programs. But really, it's a heterogeneous population of cells. You have stem cells, you have myeloid prime cells, you have lymphoid prime cells, you have erythroid prime cells, and so really this was a perfect time to start using single cell applications. And so in collaboration with Elham Azizi, who at that time was in Donna Pear's group, we did single cell RNA-seq of this stem progenitor population, shown here where in shades of blue are the STAG2 knockout biological replicates, and in shades of red and purple are the stem and progenitor cells of wild type controls. And so they do segregate out slightly differently, but more importantly, you see an enhancement of cell cycling, and then perplexingly, we saw this antipoise for lymphoid priming. Now, this is in the stem cells. Now, when I think about hematopoiesis, there's kind of this dendrogram, and you have the stem cell all the way on the left, and then you have kind of an activated stem cell, and it's not until you get 3 points out in the dendrogram that you have myeloid to lymphoid fate separation. But what was surprising here is from a chromatin standpoint, and then subsequently by a transcriptional standpoint, you already had an anti-poise relationship against a lymphoid fate. So these cells already were defective for a lymphoid fate, even as high as the level of the HSC in the absence of STAG2 cohesion. Now, this was not just a stem cell effect, this was also an effect in all 4 lineages. So erythroids were defective, megakaryocytes were defective, myeloid cells were defective, and lymphoid cells were nearly absent. So here are the UMAPs for those. We can see by pseudotime here, a delay in a erythroid maturation program, a delay in a granulocyte maturation program, and something that Elham and I in large hope to a new and very talented student, Edna Stewart, we're going to try to find out what is the major difference here in this UMAP that looks almost like a lobster claw. There's really distinct trajectories that these cells are taking over the course of maturation. And it's not simply reflective of this pseudotype. Okay, and then so nobody in this room is a hematopathologist, but I do want to highlight a few things here. One is that the megakaryocytes look like the megakaryocytes we see in humans who have myelodysplastic syndrome. They're small. The nucleus should be multi-lobed and polyploid, but it's actually rather small. And so they're not able to bud and make the requisite number of platelets. Then up here, you can see the erythroid signature actually has something we call nuclear cytoplasmic dysynchrony. So the transcriptional maturation of the cell and the nuclear condensation are no longer in sync. The cytoplasm is quite blue. It has lots of mRNA, specifically mRNA for hemoglobin. And yet the nucleus has not caught up. It has not condensed. And in something that Varun Sudanagunta, a medical student in our lab, is working up, we see quite a bit of defective erythropoiesis as well, functionally. So what we did is we took different subtypes of populations. So hematopoiesis is nice because in addition to doing the single cell technology, which we all, I think, appreciate, we also can sort out subpopulations of cells based on extracellular protein expression. And so here's data from lineage negative stem progenitors, but we also have this data from the stem, the myeloid, and the erythroid cells. But here on the x-axis of this plot on the left here is the log2 fold change of RNA-seq, and on the y-axis is the log2 fold change of ATAC-seq. And so you can see increased accessibility and decreased accessibility on the y, increased expression and decreased expression in the STAG2 knockout on the x. And the majority of differentially expressed genes fall in this left lower quadrant. So in the absence of STAG2, you have decreased expression, you have decreased accessibility. And I want to amend what I just said. It's not decreased expression. It's that these genes never turned on in the first place. So these genes fail to initiate, fail to engage, and they are all transcription factor the transcription factor that is the target of these genes is PU.1. Now, Ken Zaret has taught me that PU.1 is a pioneer factor. It should be able to bind to heterochromatin, it should be able to open heterochromatin, and it should be able to turn on these genes. So first of all, is this just a PU1 problem? If PU1 is not expressed, that would explain all of this. So we sorted cells that we know to express PU.1, and we know to express EBF1, one of the PU1 targets that was represented on that former plot. And it turns out that in the absence of STAG2, these cells express PU.1 just fine, but they failed to turn on their transcriptional target, even as a pioneer factor, which tells me that STAG2 licenses the pioneer function of PU.1. We showed this by ChIP-seq in the absence of STAG2, PU.1 fails to bind to its targets at EBF1s in cancer, as well as an intergenic site. So I told you that none of this is observed in STAG1, so we looked to see are there STAG1 specific sites, STAG2 specific sites by ChIP-seq, and we found that there's a subset of sites where STAG1 is able to rescue in the absence of STAG2, and there are sites where it is not able to rescue. And just as I alluded to earlier, CTCF, both are able to bind to just fine. They can demarcate the genomic neighborhoods, and the macroscopic three-dimensional structure of the genome is totally intact in the absence of STAG1 because STAG2 can rescue it. And in the absence of STAG2 is still able to support the three-dimensional structure at the macroscopic level, but not at loops that are targets of PU.1, IRF8, and SPI-B, which I'll come back to. So in order to look at this more closely, we measured this using Hi-C. Hi-C is a technology that was originally designed to align genome assemblies. So what is the assembly in short read sequencing of a genome? You cut it up into pieces, you sequence it, and then you find where there is contiguous reads throughout. So this dark abscissa here on a contact plot is your contiguous genome. Where you have interactions in cis across the genome, like between A and B, is the interactions and the red that is off the diagonal. The triangles at diagonals here between C and D, you can see there's darker red there. And so, in fact, we proved that the insulation score at the TAD boundaries is unperturbed in the absence of STAG2. But at EBF1, which STAG1 is not able to rescue, you now have a broken loop. Instead of showing it by a contact plot, I actually like to show the raw data better. So here is an insulation score at the enhancer and the promoter of the EBF1 locus. And at both of those sites, in the absence of STAG2, you no longer have insulation there because that loop is broken. And so it asks this question of what's the hierarchy, what's the relationship between DNA accessibility and transcription factors? And so to experimentally validate this, the first thing we did is we just expressed the heck out of PU.1. So we overexpressed PU.1 by transfecting in a cDNA that expressed it 3 log-fold higher. And then we sorted those cells and put them in culture to try to make them into B cells. And here on the left, you can see a histogram. This is for the marker B220. This is a mouse B cell marker. And so in this media, we're able to take wild type cells and really push them into B cells. Without STAG2, that simply doesn't happen. But if you add back PU.1, you're not able to get that B cell. The only thing that we learned from this experiment is if you knock down PU.1, you can phenocopy because you can't make B cells. So then the question was, what if we just overexpress EBF1? And so that we did that experiment. And when we overexpressed EBF1, which is past this chokehold of the 3D chromatin, it turns out that EBF1 potently pushes towards B cells, and it still does it even in the absence of STAG2 in vitro. It abrogates the seroreplating of the stem cell phenotype, and then finally is able to rescue B cells here in transplanted recipients. So all of that's to say that cohesin and chromatin is really important not only for transcriptional programming, but it actually works in concert with transcription factors to exert their effect. And one experiment that we're currently ongoing, which has kind of always been a thought experiment, is, well, take certain inflammatory cytokines and put them in myeloid cells and put them on B cells. It's the same signal. It's received the same way. And yet the transcriptional programs that turn on, we know that they're some overlapping, but most are different. Is the chromatin what's defining that? And so in collaboration with Russell Coons, who Simon mentioned, who's an incredibly talented biostatistics graduate student, we asked the most basic question, which is transcription factor activity associated with an increased probability of accessibility? If you think about what is a pioneer factor, right? By definition, a pioneer factor is a transcription factor whose expression and activity is able to increase accessibility. And so we would hypothesize that you could model this. And that a pioneer factor might be like this one here on the left, where you have even a small amount of transcription factors able to massively increase accessibility. We think most transcription factors are probably going to be somewhere in the middle, where there needs to be some threshold of expression in order to open access, turn on other transcriptional programs that are going to remodel chromatin. And then there's some that are going to shift to the right. And by shifting to the right, does that influence negatively what a transcription factor is able to do? And so back to this operator switchboard, the question is chromatin, by taking the 22,000 genes in the genome and segregating it into which ones are even options to be turned on, is that influencing your TF's activity? And so in order to model this, he generated something that's akin to a Hill coefficient. So modeling transcription factors and seeing if we can create a curve that represents its unique transcriptional effects and influence a relationship with accessibility. Here, where theta is the threshold by which a transcription factor is able to have 50% chance that chromatin is now open. So based on the data I just showed you, Russell and I sat down and we hypothesized two potential outcomes. If wild type is what we think the left is going to look at, where transcription factor expression is going to reach a threshold, at which point accessibility opens massively, and then will have some sort of limiting effect going on. I would hypothesize that for STAG2 deficiency, one of two things might happen. One is that you may have chromatin that is closed and simply unable to be open no matter what happens. That's what we model here in kind of the middle here. And then some transcription factors which may be turned on, but just might be slower or more resistant to opening, which would be the shift to the right as we see here. Here's the experimental data to support it. So I showed you a bunch of genes that were on that plot between accessibility and RNA-seq. And EBF1, my favorite gene, is on the left. This is not from our data. This is from human bone marrow, where single-cell RNA-seq and single-cell ATAC-seq was generated to make these programs, and then take STAG2 high expressors and STAG2 low expressors and model the patients who just normally, in terms of their random expression of STAG2, are either 2 standard deviations above or below the mean. And looking at accessibility as a function of PU.1. SPI1 is the gene that encodes PU.1. Sorry, I should have said that earlier because that nomenclature goes back and forth. But in fact, STAG2 high expressors are able to open the EBF1 locus in a manner that we would expect to see by the Hill coefficient with a transcription factor activated gene, whereas it's far more flattened when you have STAG2 low expressors. So those patients presumably have more resistance to their ability to open, turn on EBF1. By contrast, we think PAX5 is one of these genes that's just more resistant. It's not off, it's not completely closed. And there are other mediators that will help to open PAX5, but this is that right shift. And then MAC2 is something that we think is probably somewhere in the middle. So is there a myeloid-specific chromatin context that's perturbed when you delete STAG2? And is that myeloid-specific transcription factor context something that we can define in terms of a hierarchy and a dependency? And in terms of that, what is just normal myeloid differentiation, both from a chromatin accessibility standpoint as well as a TF standpoint? So Russell went back to our original single-cell RNA-seq data, and he isolated genes based on a single-cell program that represented this transition into early myeloid development. And so identified the percentage of cells that represent this stem to myeloid-committed progenitor step, and then created a UMAP that he then overlaid transcription factors to define a differentiation score. Now, the transcription factors that were key to that look very similar to that STAG2 dependency map that I previously showed you, and decreased myeloid differentiation was one of the hallmarks that we saw in the slice. So the 3 of the top 6 transcription factors that were computationally predicted to be associated with myelopoiesis were experimentally validated in our model system. So then, if there is a transcription factor dependency on the chromatin context, can we then now go back and map this from a 3-dimensional chromatin standpoint? And so this was an experiment that was done by an incredibly courageous and diligent technician in the lab, who's now a medical student at Harvard Medical School, Vesmira Aliyah. And so this is the lowest input first primary cell high chip experiment that I'm aware of. So this is a CTCF high chip of STAG2 mice in their LSK compartments and in their GMP compartment with and without STAG2, and I'm going to walk you through it here. So this is that contact plot again. And so in the middle of this track is a gene called MECOM. You need MECOM if you're going to be a stem cell. MECOM is one of the genes that gives the self-renewal potential of that cell. In kids who are recently discovered to have bone marrow failure at birth, there is a rare condition where you have a mutation in MECOM. Those babies cannot make blood because they don't have stem cells. They need a bone marrow transplant either in utero or before 4 months of age. Otherwise, they're going to die because their stem cells will exhaust. So you need to have MECOM. As you can imagine, leukemias like to overexpress MECOM, and that's a bad thing. So you need to turn MECOM off. So I'm going to walk you through how MECOM gets turned off from a chromatin standpoint first. So here in red, in yellow, there is the gene of MECOM, and there is a thin bar here which shows you the promoter. That promoter is interacting in this circle here with a locus control region, a candidate regulatory region in the non-coding region between the gene EGFEM. In that interaction, there's also an interaction with the 5' UTR, which is shown here by this flare. That creates a 2-leafed loop from the CTCF that is at the candidate regulatory element to the promoter, and from the promoter to the 5' UTR. When this cell commits to a myeloid fate, it turns it off. And CTCF disappears from the candidate regulatory element, and it disappears from the 5' UTR, but it's still there at the promoter. And as you can see here, those interactions by high C no longer exist. Now, interestingly, the stem cell in STAG2 doesn't express MECOM at high levels, but it does express it persistently. And what ends up happening here is that you still have CTCF at all 3 regions, but without cohesin, you can't tether this long-range 1.7 megabase interaction. So that's gone, but the 5' UTR still persists. So now let's dig in a little bit deeper into the promoter 5' UTR interaction here. And as you can see where you have the CTCF track in the LSK stem cell and the GMP myeloid cell, the promoter is still open and accessible. It actually doesn't change. We also looked at DNA methylation because methylation is very, the CTCF binding site is highly CG rich, and CTCF binding is highly influenced by DNA methylation. And if you look at the promoter, if you did reduced representation by sulfite sequencing, if you did a methylation array, you'd be looking at promoters. And you would say that there's no difference in the methylation of the the MECOM gene in stem and progenitor cells. And you miss the fact that all the interactions are actually happening at this differentially methylated region that is at that 5' UTR. And you methylate the 5' UTR, you evict CTCF, you lose accessibility in the GMP specifically, and now you break the loop. So that's how we think that methylation, CTCF binding, and 3D chromatin are enabling chromatin accessibility for certain transcription factors. This is a target of FLY1 to be turned on. So going back here, thinking about how this interaction between CTCF, DNA methylation, cohesin, and bringing these rings together to create the tension for expression of lineage factors also has the opposite effect, where you want to methylate certain factors, break these loops, and turn these genes off. And so we wanted to see, is that interaction actually happening? So we took the LSK stem cells, the GMP myeloid progenitor cells, and we looked at CTCF as a function of whole genome bisulfite sequencing, and we found 3 different groups. In black here is the CTCF that was at the CTCF binding site, both in the stem and the progenitor cell, and it stayed there. In blue are the CTCF that are lost and evicted. And you can see it's centered around the CTCF motif here. It doesn't take much methylation. Actually, methylating just the common 19 base pair CG-rich CTCF motif is enough to evict CTCF. And now that loop is broken and that regulatory element is turned off. And then finally, in order to tether and turn on these transcription factor programs, it actually takes about 202 base pair repeats on either side of the CTCF, which must have some localizing structure, but we don't know exactly what it is hypomethylated in order to gain CTCF and create that toggle. So let's intersect them all together. Well, I just told you that MECOM is a Fly1 target, and Fly1 targets are highly enriched for self-renewal and stemness. And what happens when you break loops? You lose anchors, you methylate, you lose CTCF, you break a loop. One of the top targets are targets of Fly1. Well, Russell told me that the most important myeloid-dependent factors are CBP alpha and PU.1, and sure enough, where you're hypomethylating the genome, now adding CTCF, creating a new toggle, and providing a loop. Well, experimentally, we validated that CBP alpha and PU.1 are the transcription factors that are driving it. So finally, the question is, how does this influence? I started by talking about the difference between AML that arises from MDS and AML that is de novo, AML that just kind of comes out of the blue. And in this altered chromatin context, what are these driver mutations doing? And so that's something that Jane Shu, a really talented postdoc in the lab, is working up and taking our STAG2 mouse models and then adding as a second hit in a sequential mutagenesis model, either FLT3 or NPM1, and how does the primary leukemia differ from the secondary leukemia that had a pre-existing STAG2 mutation for 4 to 8 weeks. And what we see is when you have a FLT3 mutation, this is your prototypic oncogene. This is, gas pedal stuck to the floor, turning everything on. You lose your long-term stem cells because they all exhaust. And so in black here, you see this directionality of FLT3 alone, and it goes to enrich both an MPP and GMP population with a highly proliferative effect. It also expands this MPP4 lymphopoietic progenitor population, which don't make mature lymphocytes normally. And that creates the leukemia, a highly proliferative leukemia. One thing that's really confused the field has been that this model of leukemia, by turning on FLT3, doesn't meet one of the criteria that experimental scientists say is essential to defining a leukemia, which is if I take those cells out and I put them in a lethally irradiated mouse, that recipient will now have leukemia, that there's transplantability. Now, of course, this is not some bar that we expect humans to reach. We will diagnose a human with leukemia based on whether they have evidence of leukemia in their bone marrow. But we obviously don't transplant it into people. And even when you transplant it into xenograft mice, it only sometimes works. But in the mouse models, one of the challenges has been it doesn't transplant. It doesn't transplant because it doesn't have the long-term HSCs that are required for durable long-term engraftment, because these cells are all pushed out of stemness and into proliferation. When you perturb STAG2, these cells come back. And I will tell you that there are 13 or more different crosses of FLT3 ITD that exist in the literature, and not a single one of them has reemergence of these long-term HSCs. So there is a transcription factor program that is defective in this exit from stemness that prevents FLT3 ITD from exhausting the stem cell population, and thus this leukemia right here is completely and totally transplantable. Now, why does this matter? Well, it matters because that long-term HSC has a quiescence to it. So this feature plot right here shows that these LTHSCs have an intrinsic quiescence that is not shared by the FLT3 leukemia alone. And so we hypothesize that perhaps this represents a leukemia initiating cell that is resistant to treatment, resistant to chemotherapy, resistant to targeted inhibitors. And as we started to put pen to paper to think about how we would experimentally approach this, it turned out we actually already had some data. So going back to a clinical trial that was run between UC San Francisco and one of the investigators, Kathy Smith there, we looked at pexidartinib. And we had done mutational profiling at initiation of this FLT3 inhibitor. And some patients fared well and were responders, and some patients were not. And we sequenced the responders and the non-responders. Every single patient that had a STAG2 mutation failed to respond to therapy. And it was the second most predictive gene that was found to segregate with disease non-responsiveness. Meanwhile, another group at MD Anderson had used a different drug, which has actually now been FDA approved, called crenolanib. And crenolanib, in every single STAG2 mutant patient in their cohort, failed to respond to therapy, and actually circled here, all increased in their variant allele frequency as they had clonal growth while being on therapy, suggesting that remodeling the chromatin landscape by STAG2 is altering the pathophysiology of transformation in FLT3. And in doing so, that quiescence, we think, is what's driving this resistance pattern. So here's how we're going to approach it, is we're going to build both a mouse switchboard and a human switchboard. In the mouse, we've now created and engineered a reversible dual recombinase mouse model using two different recombinases. Now in vivo, we can take a wild type mouse and we can turn off STAG2 just as we had done before, using a DRE recombination with ROX. Now you have STAG2 that is off, followed by tamoxifen pulsing. Now you can use a CRE recombinase to flip that point cassette back. And now in vivo, go from wild type to knockout back to wild type and see what are the reversible effects now that we've re-engineered the STAG2 to be expressed. Are there elements of transcriptional and epigenetic programming that are reversible, and which are hardwired in now that you've had that epigenetic memory set? We're also going to build the switchboard through a cohort of patients who are being enrolled in an investigator-initiated therapy of an all-oral therapy for relapsed refractory leukemia. We're going to be looking at high-C, CTCF, ChIP-seq, and single-cell RNA-seq from day 0, day 14, and day 28 of every patient who goes on this study to see how does changing the methylome alter the 3D chromatin. I would hypothesize that there might be a chromatin state that might be more permissive to these therapies. And I might hypothesize that there are certain loci that are important to predicting response and others that don't matter. So finally, I want to leave you with a couple of thoughts and point out where I think the field can go. One is that I think aberrant chromatin structure is a universal feature of many different types of cancers, particularly hematopoietic dysregulation. And in doing so, totally changes the transcriptional potentiality of the cells in which those mutations are observed. And secondly, I think that there's real potential to thinking about epigenetic targeted therapy. And there are some early indicators that we might be on to something. Certainly, I think what's been most prevalent in the lay literature are some of these exciting inhibitors of menin, inhibitors of IDH, and these drugs are hopefully starting to rewire the epigenome of these cancer cells. But I'd actually like to go back 20 years to a disease called acute promyelocytic leukemia. Acute promyelocytic leukemia, as I mentioned during the lunch today, is famous for two reasons. It's famous for one, in patients in whom it is diagnosed in the absence of treatment, the survival is less than 1 month, period. Two, it is famous because it is from a translocation between the PML oncogene and the retinoic acid receptor. Thus, the PML gene, which lives in PML bodies and the membraneless organelle of a cell, aids and is a transcription factor to turn on key myeloid programs for terminal myelopoiesis. It is now translocated and bound to the retinoic acid receptor and sequestered out to the cytoplasm. So the second reason it is famous is because if you treat with retinoic acid, also known as vitamin A, you can cure these patients. And so a combination of chemotherapy plus high doses of vitamin A takes APL patients, rewires their chromatin, now creates functional differentiation, and cures 92% of these patients. So the reality is there is real possibility to rewiring the epigenome as a therapeutic strategy. And many of these chromatin modifiers have enzymatic activity, which means they are very good targets for pharmacological intervention. The challenge is going to be which are the right targets and how do we get locus specificity. So finally, this is a really dynamic state of chromatin. I think if you look at data in single-cell RNA-seq, single-cell ATAC-seq, 3D chromatin, you're seeing a point in time. But these are dynamic changes in the chromatin. And not just cell division differentiation, but I mean changes in chromatin such that if you come across an external signal of inflammation, of a mitogen, of a growth factor, cells respond. And how a cell is primed or anti-primed to respond to a particular signal influences its intrinsic cell identity. And so as we learn more about this interaction and association between transcription factors and their accessibility, I think we're going to learn that accessibility in chromatin is its own regulatory mechanism that is non-hierarchical and non-redundant with transcription factors. And in concert, the two really define how a cell is going to function. So I've tried to name the people who have done the work along the way. Jane Xu, Yi Chen, who's here with us, has done a lot of the computational biology. Russell Coons is approaching this transcription factor, chromatin mapping and interactions together. I have a really terrific heme malignancies program here at Columbia. And I think if there's one thing that I hope to do in the next year is, as IICD begins, continues to grow, is really to try to engage from the clinical side, not only for sharing resources and clinical samples, but also sharing knowledge. To Simon for really helping to bring me into IICD and learn about the amazing research that's going on here. The Stem Cell Initiative up at Columbia with Emmanuel as the head, and my collaborators from MSK. And with that, I'll thank you for your time and take some questions.
Thanks, Aaron, for a fantastic talk for our first of our series. Sorry, I'm staring at some light. Does anyone have any questions? Actually, there are a couple online if they're still there.
Great.
Or are they?
No, it was just me asking.
Oh,
if you
okay.
have any question.
Okay. I thought somebody had pinged in the middle.
Yeah.
I don't know if this is a naive question, given how noisy it is, but theoretically, given the contact map, is it possible to recover the 3D structure of the chromosome?
Totally. Like you mean from like a macroscopic level in terms of like taking those data and making some sort of 3D model of the nucleus? Absolutely. So it all depends on the resolution, right? And so, when we think about this in space and in time, I'm showing data from cis interactions within a chromosome. But we also have interactions that are many fewer calls at interchromosomal interactions. And this is something that if somebody is interested, we really have not, systematically been able to assess. Come on, there we go. Okay. So going back to one of these graphical slides here is how can we tell, is this chromosome 1 and this is chromosome 2, right? Well, you should, because these interactions here, although they're not going to be very frequent, should exist. And they should exist at some level higher than background. It's going to take deeper sequencing. It's going to take more complex genomes. You are always at a limitation of one of 3 features in terms of looking at 3D chromatin. How many cells do you start with? How complex is your library and how representative is it of the entire genome? And then what's the resolution at which you're trying to capture the genome? If you want to call loops, you got to get down to about 5 kb windowing. And that means you really have to have a very complex library and deep sequencing. And then, technological changes that we're happy to share with anybody to try to optimize for a low input system. All right, well, this was fun.
Thank you again, Aaron.
Well, thanks for having me. This was a blast.